So I'm going to tell you about a project which is along the same lines, because we're all talking sort of about the microbiology of different crops. But in a nutshell, this project is about developing new products that might increase the yield and therefore profitability of Australian horticultural crops. And the approach we're taking to do this is analysing the microbiomes that are associated with these crops, all the way through from seed to harvest. We're also looking at how microbiomes might change when they're exposed to biotic or abiotic stresses. And the aim there is that we may be able to select some microorganisms from within the microbiome that are associated with that stress tolerance to then develop these into new products, and hopefully products that will assist plants that are under stress so they can continue to yield. Briefly, this is the team. I just want to point out the A team over here on the left-hand side. These are the people doing the hands-on work. We've got various different expertise from Murdoch University. I'd also really like to acknowledge our industry partners. So they're heavily involved in our field trials, Melville Park and Eco Growth. And then we have some advisory groups and other participants, and especially our growers from around Australia who are supplying samples for us. So the project's not possible without this huge team of people. Now, before I leap into what we're doing and the results we've got, I just want to talk a little bit about the focus that we have for this project. So we're not doing protected cropping. Our project is a field-based project. And the reason why is because there are actually very limited studies on microbiomes in field-grown crops. And in fact, most of them, when you look at them, are just snapshots in time or space. So research teams seem to go out into a field, walk through a crop and take some samples, randomly sampled, but they only do it once. They don't seem to follow it through the season. So much more detailed information is needed on field-grown crops. So that is the focus of this project. We are going out and we're sampling field-grown horticultural crops from different growing regions across Australia. And we want to unravel some questions like, how does the variety of the crop or the soil type or the climate or even the agronomic practices shape the microbiome? Does the microbiome actually change over time through the season of the crop? Or is there a stable core microbiome that is always there that the plant needs to grow? And most importantly, is there a correlation between the microbiome and crop performance? So the growth of the crop and the yield. Now, our other focus is that we want to look at the whole microbiome. So most studies do tend to focus just on the bacterial microbiome, and there is a reason for that. It's because there's a lot of data available that you can compare your own microbiome against. But probably only about 10% of studies have looked at the fungal microbiome, and yet fungi are so important for plant growth. Not just for killing them, there are good fungi as well. And there are also many other microorganisms that form part of the microbiome, including protists and archaea. So we want to look at the whole picture. So we're using both metabarcoding, so amplifying specific gene regions, and also metagenomics, which is whole genome sequencing. So we can get a big picture of the microbiome. And the more information we have, the more we will know about what are the microbes that are existing in the microbiomes. Are they beneficial? Are they saprophytes? Are they pathogens? And also, what are they doing? So the more information we have, the more we know about their function and how that influences plant growth. So in conjunction with Hort Innovation, who is one of our major funders, and also we have a stakeholder advisory group, which has industry, government, various growers sitting on it. We selected 3 horticultural crops. We've got tomatoes and avocados and potatoes. So 2 vegetable crops, both annuals, and a perennial fruit crop. And the stresses that we've identified, thanks to the advisory group, we've got 2 pathogens, Fusarium, which is common across Australia in tomato crops, Phytophthora, which is another huge problem in avocado plantations. And then for potatoes, we've got abiotic stresses, which in our instance, we've chosen heat and how calcium also influences heat stress or heat tolerance and the quality of the potatoes. And there's a picture of our beautiful student team there. So Henry's on the left, Sumitra in the middle, and Assad on the right. And most of what I'm presenting today, or in fact, all of what I'm presenting today is Henry's work. So I just want to acknowledge that. So we're going to be talking about tomatoes. Now, obviously, if the main aim of this project is to develop new products, first of all, we need to know that product application actually has some sort of, reasonable result if it is applied in the field. And there aren't actually a great deal of studies that have been performed really well with lots of replication or even repetition. And strangely enough, some of them are actually being performed by the product companies themselves. So there's no independent assessment, if you like. So we're really looking at, can we start by demonstrating product application does work? And if so, is it related to the microbiome? So Henry, to his credit, in his very first year of his PhD, has done 2 field trials on 2 separate farms, testing 3 different biological-based products applied to field-grown tomatoes. He's looked at crop growth and yield, and he's also looked at metabarcoding of the rhizosphere and the soil, both bacteria and ITS. And Anish, who is our postdoc on the project, has also taken the same samples, and he's looking at the root and the phyllosphere using whole genome sequencing, so metagenomics. So between the 2 of them, they've pretty much got every compartment in the crop covered, from the soil right through to the phyllosphere. We're not doing the air or the water, but maybe we'll do those down the track. So today I'm just going to tell you a little bit about the product application and what the results were from the trial. So this is shoot fresh weight, and this is the data that came from one farm. And I should point out, sorry, if you can hear the dog chewing on her toy in the background, I do apologise. She's a guide dog sitting there behind me. So this is on one farm. This farm did not apply any products at all in terms of conventional fertilizers or anything else. They just applied these biological products. So they either had one of 4 treatments. We had an untreated control. We had product A, which was a microbiology-based product. We had product B, which was an organic acid. And then we had product C, which was a seaweed-based product. Now, I can't tell you the names of any of those, but just trust me, that's the 3 different types of products we used. On the left, we had Roma tomatoes, and on the right we have cherry tomatoes. So 2 different types of tomatoes as well. And this is just the fresh shoot weight. I haven't included dry shoot weight here. But you can see there was quite an interesting result by applying these products. So in the Roma tomatoes, they responded very well with much more shoot biomass after the organic acid application, whereas the cherry tomatoes had a better response to the seaweed product. And that was actually consistent with the root fresh weight as well. So if you look at that here, again, same farm, same products. Roma on the left also again responded in root biomass with the organic acid. And cherry tomatoes on the right, again, had a good response to seaweed, but interestingly, the organic acid also influenced root biomass here. So what does it all mean? What we really need to know now, given that Henry's PhD is about fusarium wilt, is does applying these products and improving crop growth also confer tolerance to fusarium wilt? Now, both of the farms where we've had these trials running don't have fusarium wilt. So obviously we can't introduce it onto those farms. So for this particular part of his PhD, we're going to have to do a glasshouse trial because we can't introduce a pathogen onto a farm, as you would understand. So what we've done is we've collected the field soil from all of the plots that had product applied and the untreated plots as well. And we've brought that back to the glasshouse. He's growing tomatoes in that soil now, and he's going to infect that with fusarium. So we'll see if that soil with the amendments, which had improved growth in the plants, can actually also tolerate fusarium. Or is the soil suppressive to fusarium? We don't know. Now, the million-dollar question here is what members of the microbiome are associated with this improved biomass? And if we have fusarium, we find fusarium tolerance as well, are their microbiome, is that part of it as well? So watch this space. We are literally about 1 week away from having that data available to present, but it just wasn't ready at this point in time. But all of the metagenomics has been done. But those of you who are familiar with this technology would know that data analysis from DNA sequencing is a huge bottleneck. So in about a week's time, or maybe 2 weeks' time, ask me then, and we'll be able to tell you what the microbiome is doing as well, and if there's a correlation with that growth that we saw. So to sum up, that is our project in a nutshell. Very early days yet, but it is a field-based study on horticultural crops. We're focused on metabarcoding and metagenomics to assess the whole microbiome. And we have proven that product application does actually influence tomato biomass, which is great. Now we're looking at whether the correlation of crop performance with the microbiome may be able to lead to selection of microorganisms, which could then be developed into products. So we will keep you posted. Thank you.